Good morning, everyone.
Good morning, sir.
Good morning, sir.
Good morning, sir.
Morning, sir.
Good morning, sir.
Please answer your attendances. Roll number 66,
Yes, sir.
67, 68,
Present, sir.
69, 70,
Present, sir.
71,
Present, sir.
72,
Present, sir.
73,
Present, sir.
74,
Present, sir.
75,
Present, sir.
76,
Present, sir.
77,
Present, sir.
78, 79,
Present, sir.
80,
Present, sir.
81,
Present, sir.
82,
Present, sir.
83,
Yes, sir.
84, 84, 85,
Present, sir.
86,
Present, sir.
87, okay, 88, 89,
Present, sir.
90,
Present, sir.
91,
Present, sir.
92, 93, 94,
Present, sir.
95,
Present, sir.
96,
Present, sir.
97,
Present, sir.
98, 99, 800,
Present, sir.
801,
Present, sir.
802,
Present, sir.
803,
Present, sir.
804,
Present, sir.
805, 806, 807, 8,
Present, sir.
9, 9, 10,
Present, sir.
11,
Present, sir.
12, 13,
Present, sir.
14, 15,
Present, sir.
16, 17,
Present, sir.
18,
Present, sir.
19,
Present, sir.
20, 21,
Present, sir.
22,
Present, sir.
23,
Present, sir.
24, 25,
Present, sir.
26,
Present, sir.
27,
Present, sir.
28,
Present, sir.
29,
Present, sir.
30,
Present, sir.
31,
Present, sir.
32, 33,
Present, sir.
34, 35, 36, 37, 38,
Present, sir.
39,
Present, sir.
40,
Present, sir.
41,
Present, sir.
42,
Present, sir.
43,
Present, sir.
44,
Present, sir.
45,
Present, sir.
46, 47,
Present, sir.
48,
Present, sir.
49, 50,
Present, sir.
Sir, I'm a bit late, sir. Roll number 767.
767. Okay.
Sir, 787.
787. Okay.
Sir, 806.
806.
786, sir.
786. 786. आप तो already answer किए थे attendance. And anyone else who joined late? Okay. So in the last class, I think we were discussing something about RNA and chromosomes. Especially, we have discussed about the packing of our DNA. Today, we'll be studying about the chromosomal segregation, like during the division process. Okay. So we are going to begin with the topic called cell division. Is my screen visible to everyone?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. Okay. So we'll start with this topic. Yeah. So reproduction, what is the process basically? Why we are saying it as reproduction? Okay, it's a kind of a general English term, and reproducibility is one of the important aspects in sciences. Okay, any part of the sciences, be it living sciences or be it physical sciences, chemical sciences. Okay, reproducibility is one of the important aspects that is required. For example, let's say a man or a woman, let's say they have discovered this, they have made some invention. Okay, so when they had made this invention, they have to note down everything so that when the prototype works, next time they have to create the uh, same material several times. Okay, and this is how most of the applications or appliances comes into the existence. Okay, they are reproducible. So same way, the living cells also should be able to get reproduced. Okay, in certain cases, the living cells has to get reproduced for carrying forward the progeny, and in certain cases, they are required to take part in cellular development or tissue development or organ development and growth. Okay, like in case of bacteria, let's say reproduction, we have the cells dividing, and that's kind of a reproduction where you're seeing that the progeny is being forwarded. Okay, whereas in the sense higher creatures like us, we have two methods of reproduction. One is asexual mode, and the other one is sexual mode of reproduction. Asexual mode of reproduction, where we are actually not taking part in any sexual activities, and where the sexual chromosomes do not play a major role. Okay, so such kind of reproduction is called as asexual reproduction. So we usually see these kind of reproduction in all our somites. Okay, we have two categories of cells. Number one are somites and number two are gametes. Somatic cells. Somatic cells या somites क्या होता है? These are the cells that takes parts in functioning of all the body parts except your sexual organs. Okay, and we have another category of cells called as gametes that takes parts in sexual reproduction process and will be resulting in formation of a completely new individual. Okay, so we have types of division. We have a sexual mode of division and we have sexual mode of division. A sexual mode of division is the majority of the format of the division that usually happens in the nature. Okay, example, let's say a child is growing and from his birth till adolescent till adult and till death, the cells of his body keeps dividing. Okay, you have your blood cells getting replenished every time. You have your so-called skin cells, they worn out and then they are replaced by new cells. You have your hair, the hair keeps growing. Okay, so this way we have different types of cells available in our body which keeps on regenerating or reproducing, and that's required for organismal development and growth and functioning. Okay, so majority of the time we undergo asexual mode of reproduction, and whenever we want to produce a new baby, at that point of time, we take part in sexual reproduction process where we have to produce our gametes. Gametes. Now, looking at the gametes and somates, the chromosomes that we are talking about earlier, the chromosome number is different. Okay, in case of a regular somatic cell, the chromosome number is exactly the same as the entire what do you say body's chromosome number or organism's chromosome number. For example, let's consider we are humans. We have a chromosome number of 46. Okay, or you can see 23 pairs. Okay, so 22 pairs of autosomes, one pair of allosome. So in a somatic cell, you have all these 23 pairs, which means we have two sets of chromosomes. We are diploid in nature. Diploid. Ploid is something that determines about the number of the chromosome, number of sets of chromosomes. Okay, so if you have one set, you can call it as uniploidy or you can call it as haploid. Haploid. Okay, is the term which we generally use. If we have two sets of chromosomes, we see it as diploid. Okay, so haploid, diploid are common formats that you see in the nature. And then we can have special cases like we can have triploid, then we can have aneuploidy, then we can have polyploidy. We have different other versions of chromosomal sets availability. Okay, when we are actually looking for the division, you have to see that the chromosomal sets are carried in an appropriate manner without any disturbances. Okay, and the division process makes sure that this thing is going on well. Okay, however, accidents can happen. Okay, and errors can occur and that can eventually rise to formation of a new cell, or it can or if it is unicellular creature, it can totally evolve into a new creature. Okay, so those kind of changes are considered to be like one of the root causes of evolution of different creatures on the earth. Okay, so let's begin with the division in prokaryotes. So prokaryotes, majority of the time, the prokaryotes also do undergo reproduction by asexual method. Okay, and things to consider in prokaryotes is that we have only a single chromosome. You don't have many chromosomes to consider diploidy or polyploidy and all. We just have only one set of only one chromosome. Okay, not even a pair. It's just one chromosome. Okay, so most of the prokaryotes have only one chromosome. Of course, the genetic sequences and all are different, but majority of the cases the chromosome number is one. Okay, and number two, the chromosome is circular in nature. Okay, in our cases, we have seen the formation of chromatids, P arm, Q arm. We have seen a dyad and a tetrad. So we have seen the chromosome formation is a bit different in case of us, but in case of prokaryotes, it's very simple. The chromatin will be forming only one chromosome, which is circular in nature. Okay, and looking at the cellular structure, the prokaryote cell is very simple. It doesn't have much of the contents inside its body, so the division is fast. Okay, it is not complicated. It has only one chromosome, so the division is very fast. So it looks like the process is called as binary fusion. We will be studying binary fusion today. Okay, and in eukaryotes method, those who have nucleus and all the organelles, as the structural composition itself is complex, the process of division is also complex. Okay, so we have initially a nuclear division, which is associated with all the chromosomal content, and followed by that we have a cellular division, which is associated with the division of all the organelles and all. Okay, so this way we have different methods, and again we have to see what kind of division we are looking at. In case of somatic cells, the division is sexual mode. In case of gametic cells, the division is a bit different, where you are actually reducing the chromosome numbers to half. You have to get only single set. Okay, so this way we have different versions. Let's begin with something called as prokaryotic cell division. So first step, as soon as a new prokaryotic cell is formed, the first and foremost thing that the cell does is that it grows. Okay, first thing what it does is that it grows. So you can see the difference between these two images. You have seen a growth. So growth, what terms the growth has happened? Looking at this first two pictures images, can somebody mention what kind of growth is this? You see the first You see the image, you see the topmost figure, and you see the second figure. What kind of growth is it? Is it numbers or is it in the size? What type of growth?
Number.
No, no, no. I'm just asking about the first two images only. ये picture में we have 1, 2, 3 steps. We have a flowchart.
Size.
So in the flowchart, just consider the first two steps. The growth is occurring in terms of size. Okay, so the cellular dimensions are basically increased. So once the bacterial cell attains a particular size, okay, it has to undergo division. Now, let me ask you guys a question. Let's say this is getting grown, right? Why can't it grow indefinitely? Why can't it become a macroscopic structure? Why it exists as microscopic structure? Anyone? Anyone?
So just a single division happened. Tell you.
Let me just give you an example. Maybe you'll understand that way. Let's take a balloon. Okay, let's take a balloon and let's keep blowing the air. So आप जब balloon में air blow करेंगे, so the size of the balloon increases. Okay, the balloon is going to grow in size. Well, so as you keep on blowing the air, definitely the balloon will increase. But at some point of time, the balloon blows away. Right? There will be no more growth, and it will be like totally disturbed. Why do you think the balloon got busted? Simple physical experiment that we have been doing since childhood.
So because it reached its limit.
Yeah.
So
It is the
because the
limit.
balloon reached
The,
its limit.
the material of the balloon basically has a threshold value. Okay, so the threshold is nothing but the internal pressure that we are giving to it. Okay, so if the internal pressure exceeds the external pressure, there is an imbalance in the pressure, and this is there is a threshold for that. Okay, so once the threshold is crossed, the balloon will not be the so-called material of the balloon will not be able to withhold this kind of a pressure, and it will be bursting out. The same thing happens with our living cells also. Okay, we are made up of something called as phospholipids, dual lipid layer. Okay, so bacteria also are made like that. So when we are increasing the cellular contents, internal contents, balloon may of course we are giving air, but here actually we are increasing the viscous content, water, cytoplasmic content. Okay, so water also will be exerting kind of a pressure from the internal side, and the membrane has a threshold value, and if the membrane crosses this threshold value, it breaks and the cell can die. Okay, to avoid that, the cell grows up to a certain extent only. It doesn't grow indefinitely. The same thing happens with most of our cells. Okay, but there are exceptions where some of the cells can reach meters, like our neurons and all, and some of the cells can grow a bit bigger, like our muscles and all. Okay, so this way we have the cells growing indefinitely. Growing definitely. There is a threshold value, so they attain a proper shape and a size. Once they attain a proper shape and size, then comes the next step. What is the next step? The DNA duplication, okay, or the chromosomal duplication. So as you have to distribute your DNA into two cells, okay. So for the distribution to happen, you have to definitely multiply your DNA by two. Okay, you have to produce two. Now, how does the production happen? Look at this. This is our circular chromosome on the bacteria. Now, this has a region called as origin of replication. Replication. Now, replication क्या होता है? This is a process where the DNA is getting doubled up. Okay, we are actually preparing a replica of a DNA, or we are preparing a replica of this chromosome. So, while we are preparing the replication of the DNA, how does this get initiated? We have a region called as ori. In case of bacteria, we call it as oric. Okay. So this ori, ये region क्या होता है? Just give me a minute. Yeah, I'm sorry. So as you can see here, yeah, we have this chromosome. On the chromosome, we have a dedicated region. On the entire chromosome, you have a dedicated region where the DNA starts to divide. The starting point is called as origin of replication. Okay, origin of replication. In short form, we call it as ORI, ORI. Okay, in short form, we call it as ORI. So at this point, the replication begins. Okay, so it has a signaling content. The nucleosides are arranged in such a way that they provide a signal to the incoming protein molecules that takes part in starting the replication. Okay, so we call, for that purpose we call this point as ori. So ये origin के पास जब शुरू होता है, what's happening? We are breaking our bonds, hydrogen bonds that are available between our chains. Okay, our DNA is double helical, right? So we are going to break the bonds between our complementary bases, and we are going to unzip our DNA. So as we unzip our DNA, now the starters will come in. So starters in scientific term we call them as primer. Okay, primer is the starter. So primer क्या होता है? This is a short, very short stage of DNA which can actually bind to the complementary site and act as the starting material for the synthesis. Okay, so when you say complementary site, you have to remember the structure of the DNA. See, every time the strands run in anti-parallel fashion. So if you have a 3 here, automatically the opposite chain will have 5. If you have a 5 here, automatically the chain will have 3. Okay, and remember, the DNA synthesis never happens from direction 3. It always happens from direction of 5. Okay, which means fifth carbon से लेके शुरू होता है and ending will be always third carbon. Okay, so this is something that you have to follow. So when we are loosening up our DNA, when we are unzipping our DNA, and when we have this addition of this, wait a minute, somebody has a doubt. Yeah, किसी को doubt है क्या? I think some of you has lifted raised hand. All right. Yeah. So as we unzip it, and as you can see, we have the addition of primers, and the primers are getting added from 5 prime to 3 prime. Here also you can see they are getting added from 5 prime to 3 prime. Now, when they are adding from 5 prime to 3 prime, there is certain thing that you have to consider. You have to consider the direction of synthesis. Okay. So look at this. We have a 3 prime site here, which is free, And so the 5 prime site is beginning from this end and it can go to the free side. Well, this is good. What about this side? You don't have a 3 prime site free, right? You are getting 3 prime site somewhere free here. So your primer bind here and the direction goes here. Okay. So when your primase, an enzyme called primase, is required to join the primer to the original strand. So when the primase fixes on this, then the elongation, original polymer synthesis starts. So what are we synthesizing? We are synthesizing a polymer of DNA. And the enzyme that assists this synthesis process is called as DNA polymerase. Okay, primer, primase, polymer, DNA polymer, DNA polymerase. So we are building a polymer of DNA, and hence we call the enzyme as DNA polymerase. So how does it run? It runs from the same 5 prime to 3 prime direction. It keeps on adding all your bases, and the bases are added according to the complementary base pairing pattern. Okay, so the enzyme keeps running through the entire stretch of DNA. Okay, so when this thing happens, you can see on one of the parental strand, because of availability of free sites, the synthesis is continuous. Whereas here we don't have we have a 3 prime site here. We started the process here, and the 5 prime site ends quickly. So we have a short synthesis here. Okay. Again, what we do, we unzip the DNA. We have found another 3 prime site here. Again, we add a stretch, and that goes till here, and it stops. Okay. So in this chain, the synthesis is happening in a reverse order, and it is always happening as the chain keeps opening. So we are going to end up with multiple number of fractions of DNA. Okay, so here we are going to have a discontinuous synthesis, and here we are going to have a continuous synthesis. Okay, the reason is simple. The direction of synthesis is 5 to 3 prime, and for 5 to 3 prime, you require a complementary parental chain availability. So here the complementary availability is there, whereas here it's not there every time. It's coming at a regular intervals, and at regular intervals, we are synthesizing a short stretch. So this goes in this direction, and in this direction continuously. This goes in this direction, but it is going as a discontinuous fashion. Okay, and such kind of fragments are discovered by a scientist called Okazaki, and hence we call them as Okazaki fragments. Okay, so once these Okazaki fragments are synthesized, now once we open up the entire, look at this structure. This is basically looking like a fork. Okay, this is basically looking like a fork. So we call this area as a replication fork. We call this area as replication fork. So in a chromosome, when we are splitting up our DNA, we are going to see that we have two replication forks that have been developed, and they extend. Okay, it forms a bubble called as replication bubble. It proceeds in both the directions, and at the end, at the end point, we have a DNA which is again signaling to proteins to stop the replication process. So these proteins comes over here, and they will be separating the two last base pairing molecules of our DNA, original DNA, and this makes the formation of two new chromosomes. Okay, so this way we have formation of our DNA chains. Now one of the DNA chain was continuous and another chain was discontinuous. Now यहाँ पे synthesis complete हो चुका है. So this is leading, but यहाँ पे we still have gaps. So we call it as lagging chain. So how do we fix the lagging chain? How are we going to fill the gaps? We are going to introduce an enzyme called as the DNA ligase. Now the ligase will come here. It will be acting on these short gaps, and it will be filling up these gaps according to the complementary bases. So ligases are one of the classes of the enzymes in the nature that are required for joining two bigger polymers. Okay, दो बड़ा polymers है अगर if you have to, if you are given many polymers or more than two or more than two polymers, all these polymers are ligated. Ligation is the process of sticking up, joining up. Okay, are combined by using this enzyme called ligase. So here we are working on a DNA polymer, and hence we call it as DNA ligase. Okay, so remember the names. We have primer. So the name of the enzyme is primase. as everything is DNA, so we are calling it as DNA based things. So DNA primase, then followed by DNA polymerase, followed by DNA ligase. So once the ligase joins them up, now this is also continuous. Okay, and that's how we have production of two chromosomes. So when you are looking at the chromosome, look at the colors. Okay, here we have two darker DNA chains. Okay, and the newly synthesized chain is represented as a light chain. So when we are replicating and when your replication forks are moving forward, and when we are completing dividing the chromosomes, you see the formation of two new chromosomes. Each chromosome has a old chain or a darker chain, which is associated with a newly synthesized chain or a light chain. Okay. So same way. You see here, you have one old one, and then you have a new one. Okay. So this kind of a synthesis, where you see half of the original content is retained, it's called as semi-conservative method of replication. Okay, we are saying it as semi-conservative. We are conserving half of original. Okay, or a part of original. So this kind of a replication is called as semi-conservative method of replication. So most of the times in the nature, this is a common method that is being followed by the living creatures. Semi-conservative method of replication. Okay. So after this is done, now the chromosomal content gets distributed. Okay, and the cell membrane starts to invaginate, and at the point of interjunction, the cell membrane splits up. Okay. So while this invagination happens, you can see that the cellular content also is getting divided, and it gets divided equally, and by the time the membranes intersect, okay, the invagination intersects, everything is distributed, and you are going to get one chromosome here and you're going to get one chromosome here. So this way, you see this one, and you see this one. All these three will be looking alike. Okay. So this will be satisfying your fourth proposal of our cell theory, that new cell arise from the existing cell. Okay. So this is the existing cell. From here only we got two new cells. So when we got two new cells, you have to observe certain things, that the chromosomal content is exactly same to that of the parent. So both parent and daughter are identical. Okay, and not just that, both the daughters are also identical. They're genetically identical, structurally identical. The contents inside are identical. So they both of them will be acting like clones. Okay, they are perfect clones. So this way, from a bacteria, you're going to get its own copies through a process called binary fusion. Now, you must have understood why we are calling it as binary and fusion. Binary because every time we are going to produce only two daughter cells. Every time we produce only two cells from a single bacteria. And fusion because we are splitting the grown up bacteria and we are dividing it into two. And hence the process is called as fission. Okay. So this is what happens in the prokaryotic cell. यहाँ तक अगर किसी को कोई डाउट है तो पूछ लीजिए. Just ask me in case if you have any doubts, especially with respect to these slides. The replication, ये जो terminology है थोड़ा बहुत different है. Okay, remember this terminologies. Origin can be shortly called as ori, and termination of replication. This is the region where the replication gets ended up. Okay, this can be shortly called as ter, ter. Okay, so ori, ter. Okay, probably you may not have a doubt with this slide, but I expect some queries from this particular slide, especially with respect to this discontinuous stands and all. Synthesis pattern, अगर आपको समझ में आ जाएगा, it will be clear. But if you are stuck, let me know. If you're stuck with any of these formations, let me know. Anyone,
Sir,
anyone.
sir, can you
Yes.
explain DNA replication also? This slide.
Yeah, this one. Yeah,
Yes, sir.
yeah, sure. आपको डीएनए का स्ट्रक्चर पता है ना? It is double helical, right?
Yes, sir.
Yeah, DNA is double helical. So it is basically having two chains. So when we are constructing a DNA molecule, when we're constructing a DNA molecule, we have earlier seen that the orientation of the sugar should be opposite. So the chains will be running in an opposite fashion. Okay, if one is going from 5 to 3, the opposite one will be coming from 3 to 5. Okay, so that's how the so-called synthesis, that's how the base pairing happens between both the DNA chains. Okay, so now once we are base paired, once we have a fully established DNA, what we have to do, we have to replicate it. So when we have to replicate it, the primary thing that we have to do is that we have to isolate them. Okay, you have to isolate the chains. So we are breaking the hydrogen bonds. Okay, so the mechanism is very complex. So we are not dealing with that. You just remember that we are breaking the bonds. You are separating the DNA chains. Okay, so once while we are separating the DNA chain, at that point only, we are adding the material that is required to start the replication. Okay, so this material initiates the replication process or begins the replication process. So how do we begin with? We begin with a very small stretch of DNA, and we call this as primer. Okay, what are we calling it as? Primer. So primer is a short stretch of DNA that gets fixed onto the parental strands in the direction of 5 prime to 3 prime. Okay, so here we have a 3 prime site, so automatically the binding of 5 prime is feasible and it gets fixed up easily. Here the 3 prime site is not completely free, it's free somewhere in the middle of the open chain. Okay, so we start our process straight away from here. Wherever there is a feasibility, we are beginning it from there. Okay, so now the synthesis of this chain will go from 5 to 3. So this is going in a downward direction, and the direction is same as the direction of breaking the bonds of the DNA. Okay, so there is no disturbance for this chain. Whereas this one, the direction of synthesis is in opposite direction. We are going opposite to the direction of opening of the chain. So the chain is opening this way, and we are going in this direction. So the synthesis will be halting after a short gap because of lack of so-called the parental chain. Okay, and uh, as we unwind the DNA, as we unwind, I mean, as we keep on increasing the replication fork size, okay, you see that one of the chain is getting synthesized continuously, and the other chain will be synthesizing in a discontinuous fashion. So why discontinuous? Again, as we open this chain, we are finding some 3 prime site here. So our 5 prime new molecule start replicating from this direction. So we have a small synthesis here. Again, we have another small synthesis here. So this way, once we split up this entire DNA chains, on this one, we are going to have a completely new, completely and continuously synthesis chain. And on this one, we have incomplete and discontinuously synthesized chains. Okay, so we are going to have a segment here, we are going to have a segment here, here. So this way, we are going to have multiple segments. So all of these multiple segments now they have to get fixed up. They have to become continuous. So to make them continuous, we are bringing another enzyme called DNA ligase. So as per the complementary basis available here, the ligase will be fixing up your DNA and it will be pasting the segments. So this way, all the entire segments are fixed up, and this is also made continuous. Okay. So once this is made, so while we are making this, you see that this is already done. This is leading, but this got lagged behind of because of this short gaps, and hence we call this as lagging chain. Okay. However, at the end, we are going to have a newly synthesized leading chain and a newly synthesized lagging chain. So this newly synthesized lagging chain will be having the sequence same as this one, okay, and this one will be having a sequence same as this one, okay. तो इसका कॉपी यहाँ पे बना है और इसका कॉपी यहाँ पे बना है. So इस तरह हम एक अलग सा क्रोमोसोम को तैयार कर रहे हैं. So we have prepared a chromosome here and we have prepared one chromosome here. Okay, so both the chains are now available in your newly synthesized uh, newly synthesized DNA. Okay, so sorry, both the chromosomes are now available with newly synthesized DNAs in them. Okay, and when we are synthesizing, we have seen that you can see one is taking blue and one is taking the brown. Same way here we are having one blue and one brown. So the original versions out of the original, let's say these two are the original persons, and let's say these two make 100% of the chromosome. So out of 100% of the chromosome, 50% of the chromosome is new moving into the new cell. Same way, this other 50 is moving into the new cell. So as we are sharing only half of it, I mean, as after the replication, the original content from the original content we are sharing half. We call this kind of a replication as semi-conservative method of replication. Okay, so semi-conservative, conservative, एक तो retaining some older parts, but semi-conservative, we are retaining older parts, but not in full. We are retaining something in certain percentages. Okay. Here, in case of the DNA replication, we are retaining it as 50%. Okay. Because 50%, this 50% is moving into this one, and the other 50% is moving into this one. And these two, they get segregated and they get occupied in different daughter cells. Okay, and that's the reason why we call it as semi-conservative method of replication. Yeah. So that's how the DNA replication happens inside the cell. All you have to remember is the direction of synthesis, okay, and formation of the replication fork, the name of the enzymes. You just remember three enzymes. एक enzyme का नाम primase है, एक enzyme का नाम polymerase है, और एक enzyme का नाम ligase है. Primase is required for fixing your starter at the beginning, followed by polymerase is required for adding all the nucleotides and making it a chain or making it a polymer. And finally, ligase is required for filling the gaps that we have found on filling the Okazaki fragments, okay, or making the Okazaki fragments continuous. Okazaki fragments are nothing but these are the same discontinuous fragments that we got during the DNA replication. Okay, so this is all about our DNA replication. And moving ahead, eukaryotic cell division, we will be studying it later. Yeah, this one we'll be studying it later, probably in the next class. Okay, so we stop here with binary fusion only. So in binary fusion, if anyone has any doubts, let me know. Okay, the process usually looks simple, but when it came to DNA replication, you have to explain a bit. Okay, I mean the chromosomal division part is crucial. So if it is coming as a short form of question, you don't have to explain about all these aspects. DNA replication में जो sequence of events था, वो explain करने की जरूरत नहीं है. Okay, but if it's coming as a long answer question, then you have to explain everything, beginning from the cellular growth till the end. Okay, so we stop here, and people who have given attendances they can go. People who have to give the attendance, they stay back. Thank you all.
Thank you,
Thank you,
sir.
sir.
Thank you.
Thank you, sir.
Thank you.
Sir, 812 present.
812.
Thank you,
Okay.
sir.
769.
812. Yeah. 7.
Sir,